your eyes closed. And polysomnography, or PSG, it is known as a sleep study. It is a test used to diagnose sleep disorders. It records your brain waves with the help of electroencephalogram, your oxygen level in your blood with the help of pulse oximeter, and your heart rate with the help of electrocardiogram, breathing during sleep. It also measures eye and leg movements with the help of electrooculogram and electromyogram, respectively. So, what was exactly the study objective? Quote unquote, in young adults, napping is hypothesized to benefit episodic memory retention, example via consolidation, to confirm and to check whether this relationship is present in older adults, as it has not been adequately tested. But it is an important question because older adults display marked changes in sleep and memory. So the design was between subjects design. The setting was in sleep laboratory at Emory University School of Medicine. Participants were 50 healthy young adults in the age bracket of 18 to 29 and 45 community dwelling older adults, 58 to 83 age bracket. Intervention was participants were randomly assigned to a 90-minute nap opportunity or an equal interval of quiet wakefulness. Definition I have mentioned earlier. So what was exactly the design of the experiment? So 2 sessions were held, session 1 and session 2. Session 1 mainly focused on the screening part, that is to screen for patients having sleep apnea, dementia, determine whether the person could nap during the daytime in a sleep laboratory, self-reported history of psychiatric, neurological or cognitive disorders, use of medications that affect sleep architecture. So the screening process was very important. So first they did was the screening part. Secondly was the session 2. It was a 90-minute session where the participants, it is 50 adults and 45 young adults and 45 older adults, out of the total 111, that is after the screening process, were instructed to firstly remember, it was written in green font, and to forget in red font individual words. They then viewed 100 words sequentially. So if you look at the picture on the right hand side, here actually, for example, if the word is fiscal, it was shown on the screen for 3000 milliseconds. Then a blank screen was shown, 500 for 500 milliseconds. And then this remember or this forget screen was shown for 2000 milliseconds. And then the patients or the participants were randomly assigned to a 90-minute nap opportunity or a 90-minute quiet wakefulness. So moving on to what was the exact measures that were taken, like in between the experiments and also before the experiment. So firstly, we know that caffeine, nicotine and alcohol, they affect our sleep schedule. So participants were instructed to avoid such things on the day of their study. A practice session was held where again the older adults were given a second opportunity to study and recall these words. Firstly, with the young adults, they were given the words on the screen, it was displayed. And second time also, they were given that opportunity to avoid age-related problems that may arise. Thirdly, EEG, electroencephalogram, was there to confirm absence of sleep in the PSG setup, a polysomnogram. It is already present. And fourthly, participants in the nap condition, they were not immediately waken up. They were allowed to sleep until their first spontaneous arousal. So, what was the result and implications? For that, what they did was they generated a polysomnographic data with the help of polysomnogram, and they also did an additional statistical analysis. So, if you see on the left side, I have it is directly taken from the paper. They mentioned that we used t-tests, student t-tests, analysis of variance, ANOVA, and the analysis of covariance to determine whether condition, nap, quiet wakefulness differentially affected memory performance in young and older adults. When necessary, we use Levens correction for unequal variances. We report Cohen's d and partial eta squared as measures of effect size. So if you look at the table, it is generated by the polysomnogram or the polysomnographic data. Here, if you see the sleep efficiency in young adults, if you follow with the pointer, it is much higher compared to older adults, the sleep efficiency. And the parenthesis values are the standard deviation values which they have calculated. And here they have mentioned that the pulse oximeter data were missing for one young adult participant and one older adult. And this R values are the Pearson R values between the session 1 and 2, the polysomnographic variables. And the last column, that is the P value, the probability values for the main effects comparing young and older adults for experimental session 2. So these are the data that they have generated with the help of the statistical analysis. Now, the actual result was actually not only with the help of ESG and statistical analysis, but they also generated the results by analyzing with the help of two additional procedures. That is, firstly, this post-NAP or wake free recall performance and post-NAP or wake recognition performance. So what they did was, after that 90-minute nap session or quiet wakefulness, the patients or the participants were instructed, they were given 5 minutes to write down all the words they studied, regardless of the original remember and forget instruction. And it was scored as hits or false alarms. And the second was post nap or wake recognition performance, where they recognized the 100 old studied words which they were shown, and the 100 new earlier words were displayed on the computer. And this was calculated with the help of Levin's correction, as well as here D prime analysis also. So if you see here in the right hand side corner, they have generated a graphical representation of this recall performance. So as I said earlier, that the participants were addressed to a 90-minute session of nap and quiet wakefulness. So if you see here, the blue portion is the nap the indication, and the red is the quiet wakefulness. So here the hits and false alarms. The hits refers to words correctly recalled, and false refers to the words that were generated on free recall test, the false words that were generated. So here the hit rate was greater following the nap rather than following quiet wakefulness. So if you see the graph here, the young adults hits were the correctly word recall, so it was greater. The range was greater compared to the false alarms, and in older adults it was comparatively equal. So here actually it mentions that the hit rate was greater following the nap rather than following quiet wakefulness in young adults, but not in older adults. And the false alarm rates did not differ significantly across conditions in young adults or older adults. So you can see that in both of them, the graph of false alarms are more or less quite similar, but in case of hits in young adults and older adults, they are very much different. So the second that they did was recognition performance. This was the data that they have generated. And here, if you see, with the help of the D prime analysis, they showed that the older adults performed statistically as well as the young adults. So here you can see that the remember words, the proportion is quite, the value is quite similar, like 0.69 and 0.69, and the standard deviation value 0.12 and 0.14. They are more or less close to each other. Secondly, there was significantly better recognition performance for remember items and forget items. So here, if you now compare these two values, 0.69 and 0.49, they have very much gap difference between them. So the remember words, they were recalled quite efficiently. And thirdly, furthermore, the directed forgetting effect was larger for the young adults than for the older adults. So the forgetted words, that they were instructed to forget certain words. So those proportions were also less recognized by the young adults, and they were more recognized by older adults. You can see the value 0.59, and here it is 0.49. So they had difference, but there was a very great difference between them. And lastly, these age-related encoding differences, to minimize this, they did this thing twice in older adults and once in younger adults. So, and minimizing age-related retrieval differences by using a recognition test. So, this recall performance was more than enough, but they did a recognition performance just to avoid, in case if there is any age-related issues. But still, there were no evidence for napping benefit episodic memory consolidation in older adults. So, in other words, even when controlling for other known memory deficits, sleep still benefits memory less in older adults than in young adults. So, as I have mentioned earlier, that the recognition performance was just done to avoid confounding age-related deficits in consolidation. So moving on, as to the limitations, as we know, the research topic itself is a very familiar yet unfamiliar topic. So they had many limitations to point out. Firstly, if stabilization against, firstly, sleep and memory research has attempted to isolate the effects of memory reactivation versus stabilization of memories from interference. Both of these interpretations have some limitations when their influence are considered separately. So if stabilization against interference was the only factor promoting sleep-related benefits, then one would predict that both the young and old adults they benefited from the nap, but that was not the case in this experiment. If selective memory reactivation were the only important factor, then one would expect more pronounced evidence for retention of remember items over forget items. Yet this was also not what has actually happened. We have seen earlier in the results. Thirdly, age-related neurophysiological deficits may block memories from being consolidated even during high-quality naps. Sleep cognitive and neurophysiological mechanisms deserve further research to inform why memory consolidation declines with increasing age. So one more additive limitation of the current study was that they expected, given the 90-minute retention interval, that our methodology did not require participants to learn words to the 80% criterion. Like they had a very low recall for forget items. So this 90-minute retention interval may be not good enough to display such results, maybe, according to them. And lastly, the last fly in the ointment was the lack of significant positive correlations between PSG measures and memory retention. So here, this picture is actually nothing but the setup of the polysomnogram, which records your blood oxygen level, breathing event, and the sensors are attached in your brain to record the brain activity waves. And these are the data how they generate through a polysomnogram. So this image does that. And lastly, the conclusion was it was found that afternoon naps did not benefit episodic memory in older adults. An afternoon nap showed no significant differences relative to a nearly equal duration of quiet wakefulness for retention of strongly encoded or weakly encoded memories or for recognition of studied words. Thus, we can conclude that simply gaining more total sleep time is insufficient to produce memory benefits in older adults. And the most important thing which binds us to the course which we did, by contrast, their findings were compatible with the view that age-related changes in sleep physiology, cognitive processing, or other relevant underlying neurophysiology preclude an afternoon nap from being as strongly restorative for episodic memory in older adults as in healthy young adults. Lastly, I would like to rest my presentation. Thank you.
Thank you, Pragya. Any questions from the panelists who are here today?
Hi, Pragya. Nice presentation. Um,
Thank you.
my only thing was that in the results section, you have displayed all the results in a single slide, which makes it a bit difficult to follow. Um, I
So,
mean,
yes, continue, sir.
yeah. So if you would like split the results in two slides, maybe, and like highlight stuff that makes it easier to follow. That would have been better. Otherwise, a nice presentation, and just avoid writing so much text. Just give one or two lines of text, and as you already told most of the stuff, you don't need that many sentences or words in the slide itself. Otherwise, it's a good presentation. Yeah.
Thank you, sir. So actually, the thing is, this paper is itself a very familiar yet unfamiliar paper. So splitting the result in two PPT, like two slides, would be difficult because the results are actually interlinked. Like they did not clearly have any particular result as such in
Yeah.
case of the experiments that usually are done as a research paper experiment. But in
Mm.
this case, it had to be, actually clumped up into one slide. So for that thing, I think it may be a bit congested. Okay, sir. Thank you, sir. I will keep it in mind.
Yeah, it was a nice presentation. Thank you.
Thank you, sir.
Anything from your side, Orko?
Yeah, I had the same thing. Like only a few slides were a little crowded, but overall, and a few of the slides contained a bit more texts. Otherwise, it was a very nice presentation. Like we could get the conclusion of the paper and everything. So it was nice. Yeah.
Thank you, sir.
Okay, Pragya, from my side, very, very well structured presentation. First of all, you have really actually kept everything that we need to know. You have kept everything in your slide. You have structured it pretty well. Other than that, as you might be knowing, while giving presentations, at least in online platforms, it's okay if you have a little less font size, but if you give it somewhere, outside, you have to present it in a board or something, then the some of your
Yes, sir.
the
The,
sizes are very small.
yes,
Not
sir.
I mean, the text anyway is more, but the size is also very small. It's very difficult to follow that. Apart from that, regarding a few questions, what do you mean by sleep efficiency?
Sleep
You have
efficiency
mentioned.
is, yes, sir. Sleep efficiency was actually, they measured it with the help of the polysomnogram. Here I have shown the picture here. So here
Uh-huh.
actually there was a reading with the help of polysomnogram. A reading is data is generated where I said earlier electroencephalogram. I guess yes, I
Mm-hmm.
mentioned that.
Mm-hmm.
With the help of that, they were actually measuring the sleep efficiency. Like they calculated with the help of these maybe a t-test, or I don't know exactly, it was not mentioned, but they calculated it. But it was, the data was shown in the polysomnogram where the sleep efficiency was pictured. And they calculated with that data, like this sleep efficiency proves that in young adults, avoiding other factors that affect related to age related stuffs, young adults had greater sleep efficiency than older adults, like 54.17, 69.80. So there is a difference, gap between the value. So in that way, they wanted to mention that sleep efficiency was more for young adults than older adults.
That being said, that is one thing. Another thing, you are in this same slide, you have Pearson R value, and that correlation is between adaptation and experimental now.
Um, no, session 1 and session 2. So session
Okay.
1 was yeah. So session 1 was this. Wait, one second. Right. So, yes, session 1 was the screening part where they did that data analysis where they screened the patients with those who have sleep apnea and dementia. And session 2 was this the whole process, that nap and the quiet wakefulness. 90-minute session was provided for them. That session 1 and session 2, between these two.
So your Pearson R values are very less actually. They are very, very less.
Yes.
So what
For
what would you signify with that kind of a result? At least I can say for older adults, it's very less than that of young adults.
yes, sir.
Uh,
So they, what they wanted to mean is that just because the data was very less in case of older adults, so the efficiency, like the this efficiency was also lower for them. Like for example, here actually these all the analysis is what I did. It was not mentioned in the paper. It was just mentioned in sentences and words. So that is the reason why I had to include more and more text data. But the thing is, as the data, as the value was less in older adults, so it proves that they age related factors are present. Before even starting the experiment, they analyzed one thing, that age related factors are present. But if if at all throughout the experiment, if that can be overcome, they were trying to do that, but ultimately it was concluded that no, it cannot be done. So I think in general this explanation can be said.
Okay, okay. Thank you, Pragya. Thank you. I think what we usually say that sleep heals everything, that is not true completely.
Absolutely not, sir. Absolutely not, sir. Sir, maybe for us, sir, it is quite good, but in older adults, it is not quite not, sir.
Okay, okay. Thank
I
you
think
very much.
with the
Yeah,
sleep efficiency thing, I think it's is it like how often subject is having a deep sleep or a
yeah. I
like
think they it depends upon the length
Sir,
of REM cycle, I guess.
no, no, sir. So one thing I would like to mention is that there are two aspects of sleep. Firstly, one thing is when a person is tired or needs rejuvenation or some energy, that sleep is different. And this whole experiment was done to, check whether that retention capacity is there in young adults and older adults after that nap or quiet wakefulness or not. So these two aspects are totally different, sir. So if a person in older adults, it's not that through this experiment we will say that, no, don't sleep. No, no, not in that sense. But the thing is that they should, if they are tired, if they are stressed, they should. Everyone should. But in this case, in this experiment, they wanted to check that whether this retention thing, like remembering things after maybe a nap, 90-minute nap, or a 90-minute quiet wakefulness that is just lying with your eyes closed, like that helps in which case, like it in younger adults or in older adults. So it proved that no, younger adults have more advantage in this case rather than in older adults. So these two aspects are, I guess, totally different, sir. So, yeah, I
Thank
think
you, Pragya. Thank you very much. I think even another thing would have been better, not in your sense. I mean, in with respect to the paper, would be to see whether sleep deprivation cause any issues with remembering. But I think their objectives are pretty much
yes,
different.
yes, yes,
Huh.
yes. So actually,
Any
they did certain research was done. Actually, some research was done. From there, they try to find out the loopholes and they try to correct it here also. But eventually, there are still limitations and loopholes that are present. So, anyways,
okay, okay. Thank you very much. We'll
so,
move on to the next one. Anybody else who would come forward?
Yes, sir. I
Ingeet,
can.
okay. Ingeet or Srinita, who would be giving first?
So can I, because Shreyushkar is not here?
Okay, you can go ahead then. Ingeet, we will have you next.
Okay, sir. Actually, I and Anushka are presenting together, sir, in a team.
Can you give it after Srinita? Because she was trying in the first round also.
Okay, sir. Okay, sir.
Okay, Srinita, please continue.
Just 1 second, sir.
Yeah, sure.
Sir, is it visible? I don't know.
Sorry?
Sir, is the PPT visible?
No, it's not.
Sir, Ingrid can go. I'm having some problems with the PPT sharing.
You're having some problems. Maybe you can share it with one of us and we might,
Sir, can I share it in the group then?
yeah, in the group, you can give it to me. It's up to you.
Yeah. Okay, sir. I'll just forward it to you then. Sir, I just forward it in the group.
You forward it on the group. Okay, I download it from there. Just wait for a moment. I think you can also control it now.
Yes, sir. So can I
Okay.
start?
Yeah, yeah, sure.
Uh, Good afternoon, everyone. I'm Srinita. I'll be presenting this PPT on the topic of hallmarks of aging. Aging, as we know, is inevitable in living organisms. By aging, we mean it is the loss of physiological integrity. Physiological integrity, as in the physical health or wellness we need by providing care. The hallmarks of aging refer to concepts in the field of gerontology and biology that outlines various processes and factors believed to contribute to the aging process at the cellular and molecular level. There are, as of now, nine hallmarks. Um, the first one, the genomic instability. This hallmark encompasses DNA damage and mutations that accumulate over time, leading to cellular dysfunction and aging related diseases. The next one, the telomere shortening or attrition, which is the protective cap on the end of chromosomes, and their gradual shortening is associated with cellular senescence and aging. We have the next, the epigenetic alterations, which is the change in DNA methylation patterns and histone modifications, which can influence gene expression and contribute to age-related changes. The loss of proteostasis is the age-related decline in the maintenance of proper protein folding, and degradation can lead to the accumulation of damaged proteins and aggregate contributing to cellular dysfunction. Regenerative sensing is the altered signaling pathways related to nutrient sensing and metabolism. The next one we have is mitochondrial dysfunction, which is the declining mitochondrial function which is responsible for producing cellular energy, which can lead to increased oxidative stress and age-related damage. The next, cellular senescence, accumulation of senescent cells which have ceased dividing and can promote inflammation and cause dysfunction, is one of the hallmarks of aging. On the next slide, we have the stem cell exhaustion. It is the regenerative capacity of tissue-specific stem cells can repair tissue repair and maintenance. And the next, the last, we have actually another hallmark, which is kind of the sum of everything. It is the tissue regenerative and repair deficits. It is the age-related impairments in tissue repair and regeneration that can lead to functional decline in various organs and systems. As to conclude, these 9 or 10 hallmarks are divided into three categories. That is the primary, the antagonistic, and the integrative hallmark. The primary hallmark is the negative attribute, which includes DNA damage, having aneuploidy, telomere loss, and others. For antagonistic hallmark, wherein at low level have beneficial effects, but at high level becomes dangerous. This is the case for senescence, protecting organism from cancer, but in excess can promote aging. The last, the integrative hallmark, this is the stem cell exhaustion and altered intercellular communication which affect tissue homeostasis and function. To conclude, to conclude, the hallmarks of aging represent a framework for understanding the complex and interconnected processes that contribute to aging at the molecular and cellular levels, as I said before. While research in this field continues to evolve, these hallmarks provide valuable insights into the mechanisms underlying aging and age related diseases. Understanding these hallmarks is crucial for developing interventions and therapies to promote healthy aging and extend human lifespan in the future. Researchers are actively working to uncover new hallmarks and develop strategies to target them with the ultimate goal of improving the quality of life for aging populations. Thank you.
Okay, Srinita. Anyway, this was already discussed in detail over the entire course. So I don't know why you took up this. We already discussed this very paper also.
Yeah,
But
actually, it was not mine. Like I actually joined this group after Shreyas took the topic and wrote it in the like sheet already. So we had to go on with this.
Okay. Okay. Anyway, lot of texts here and there. It's difficult for us in that sense. Very less information. Your slides are black. I don't know why you chose that color, but it's not really very appealing. Like any color above black is not that good except white. So you have the white, but you also have green, which doesn't look that good. I mean, these are some presentation, information, other slides also. I mean, the colors, they really do not come up that well in a black setting. So yeah, that's all from my side. Anybody else? Anyone
Yeah,
else?
I think it's the same. It's just too much text, and even with the text, the font size is a little small. It's difficult to appreciate. And actually, I had one question though. You mentioned in one of the hallmarks, like it's altered intercellular communication. So can you give some examples? Like, what do you mean by that, and how does it
Uh,
affect
can
senescence?
you repeat the question? Altered. Uh, can you please repeat the question?
Yeah, I was asking that, like, what intercellular communications are altered. Basically, some examples like what happens and like how does it aid in senescence, or why does senescence increase because of that? Like any example?
Uh, so in altered cellular communication is like refers to the changes or disruptions in the way cells communicate with each other. So, gene, including the gene mutations, diseases or like diseases as not just anything specific, but the mutation, the genetic mutations that there is.
Okay, okay. Yeah, okay. That's pretty much from my side.
Actually, I'm really sorry for this PPT. I'm actually not really good with like technologies, and I'm trying to be better. Anushka here knows how bad I am with technology, gadgets, and everything, but I'm learning.
Yeah, I think we all have been novices in this at some point. Only Agradeep has to used to give very good PPTs right from the beginning. Agradeep, anything you have to say?
You pretty much summed it all. A few suggestions that are very similar to yours is that avoid having black backgrounds. Never have black backgrounds. Whenever you are
Okay, sir.
presenting, just give a plain white background is much better than a black. And also, I think your PPT would have been much more intuitive if it just, let's say, in this particular slide, you just give stem cell exhaustion and you give an image from any particular paper, and you just give like 2 or 3 different bullet points how stem cell exhaustion actually affects it. It would have been much more interactive in that aspect. But yeah,
Okay,
it's
sir.
good that you are trying to improve. Just yeah, that's all from my side.
Thank you, Srinitha. I think Ingeet will take over. Okay, I have to stop sharing it with yours. Ingeet, are you there? Ingeet or
Yes.
yeah,
Yes, sir. Is it visible?
just give me a moment to see that. Yeah, it's visible.
Okay. Good afternoon, everyone. Today we would like to present on the research paper Early Time Restricted Feeding Improves 24-hour glucose levels and Affects Markers of the Circadian Clock, Aging and Autophagy in Humans by Jamshed et al. These are the contents we will cover. So what is TRF? TRF, or time restricted feeding, is a form of intermittent fasting in which there is a longer fasting period. In TRF, we restrict the feeding time to less than 10 hours and fast for the next 14 hours per day. In humans, TRF has reportedly improved weight loss and cardiometabolic endpoints such as insulin levels, insulin sensitivity, and blood pressure when participants ate early in the day, but worsened cardiometabolic health when TRF was done late in the day. These observations are suspected to be in line with the circadian systems. Here, they extend analysis from the 4-day trial on over 11 overweight adults to perform the study on how TRF affects selected hormones and expression of certain genes. They hypothesized that eTRF would decrease mean 24-hour glucose levels, positively impact hormones such as IGF-1 and BDNF-1, alter circadian clock gene expression, increase autophagy, and have anti-aging effects in humans.
Here's a quick overview of the experiment, where we have the materials needed and the parameters monitored. As materials, they needed participants aged 20 to 45 years and body weight 68 to 100 kilograms with a regular bedtime. They were excluded if they had any chronic disorders. Participants had to wear a continuous glucose monitor at all times on the fourth day of the experiment. Suitable materials were used to analyze levels of expression of various genes and hormones. The Satterthwaite method was used for statistical analysis. The parameters monitored were 24-hour glucose levels, glycemic markers, lipid metabolism, gene expression, hormones, and effect on aging.
The experimental design was as follows. On days 1 and 2, participants followed their assigned eating schedule on their own and were instructed to maintain their habitual sleep and exercise habits. On day 3, breakfast, lunch, and dinner were served at 8 hours, 14 hours, and 20 hours, and for the control schedule, and at 8 hours, 11 hours, and 14 hours for the eTRF schedule. On day 3, they were asked to refrain from caffeine and exercise. On day 4, participants wore a continuous glucose monitor. Blood samples were collected in the fasting state at 20 hours. On day 5, blood samples were collected at 7:30 in the fasting stage.
The observations of the experiment were as follows. Mean 3-hour glucose levels were 3 plus minus 1 mg per dl lower after breakfast, but unchanged after lunch and dinner. Overall, eTRF reduced 24-hour glucose levels by around 4 mg per dl. As for glycemic markers, eTRF lowered fasting glucose and insulin by around 2 mg per dl and 2.9 mu, respectively. eTRF also reduced glycemic excursions as measured by MAGE by around 12 mg per dl, and there was an increase in IRS2 and AKT2 gene expression. As for lipids, wait a second. In the morning, eTRF increased cholesterol by around 9 mg per dl, but did not affect levels of triglycerides or free fatty acids. As a result, total cholesterol was elevated by around 10 mg per dl. As for hormones, eTRF did not affect any of the hormones in the morning except cortisol, which tended to increase by around 1.5 mg per dl. In the evening, eTRF reduced cortisol levels by around 1.4 mg per dl and tended to increase BDNF levels by 2.46 nanogram per milliliter. In gene expression, eTRF significantly increased the expression of the circadian clock genes BMAL1, CRY1, CRY2, and RORA in the morning. In the evening, it decreased levels of PER1 and tended to increase levels of CRY1, CRY2, REV-ERBA, and RORA. eTRF also increased levels of SIRT1 and LC3A in the morning and increased levels of MTOR in the evening. The autophagy gene ATG12 was elevated in the evening, but this effect was no longer significant after adjustment for multiple comparisons. The expression of all other genes was unchanged. A single bout of breakfast skipping changes the postprandial expression of several clock genes in whole blood, suggesting that a tightly controlled bidirectional feedback loop exists between meal timing and the circadian system. Both SIRT1 and NC3A were upregulated in the morning before breakfast, while MTOR was upregulated in the evening. The increase in SIRT1 expression in the morning suggests that ETRF promotes longevity in humans, like in animals. A recent study in rodents found that at least 40% of the lifespan extending effect of caloric restriction could instead be attributed to TRF. ETRF increased LC3 expression by 22% in the morning at the end of the 18-hour fast. LC3A encodes an essential structural component of autophagosomal membrane. Increasing autophagy may have anti-aging or rejuvenating effects. Other studies on intermittent fasting conclude that several of the benefits of intermittent fasting are mediated through enhanced autophagy.
Although the study was highly rigorous in design, the sample size was small and several of our endpoints were likely underpowered. Substantial decreases in IGF-1 and IGFBP1 and increases in NOS3 gene expressions, which would provide further evidence, did not reach statistical significance. Thus, these targets need to be retested in larger trials for more accuracy. CGM data was not well calibrated. Study intervention was only 4 days, which may be insufficient for circadian and or metabolic adaptation to occur. With the exception of glucose, all endpoints were measured only 2 times a day. We lack data on endpoints in the postprandial state and also while sleeping. Some endpoints such as autophagy are likely maximally upregulated during sleep. Although the PM blood draws were taken a 6-hour fast in both arms, there were differences in cumulative food intake, which may have impacted the endpoints measured in the evening. They were not able to draw blood at multiple time points throughout the day. Gene expression data are known to be limited in nature and not necessarily reflective of changes in the protein levels or activation. These are the references we have used, and this is our team. Anushka Sharkar. I am Anushka Sharkar.
And I'm Ingit Pal.
Thank you.
Anything from the audience or from the panelists?
Hi. Can you just go to the slide number 8, the observations? I just wanted to have a look. So, okay. One suggestion would be to, because there are these so many genes and everything, you can just give a kind of a condensed view on what gene affects what or what these does.
Yes, sir, done that. In that picture there is like circadian clock genes. The genes are listed below, and under longevity and autophagy genes similarly they have been listed.
Yeah, I just when you are presenting, just spend a little bit more time on these slides, because there are so many things and I was just trying to follow and then you quickly moved on, but otherwise it was a nice presentation and yeah, I liked it.
Okay, sir, we will keep that in mind.
Orko?
Yeah, from my side, it was a very nice presentation. And just regarding the observation slides in like slide 7 and 8, as I can see, so try to put the graphs or pictures a little bigger than the texts. You can always put one observation and one legend per slide. That also works. So try to put that like figure or the graph a little bigger than this rather than the text. That's all. Otherwise, a very nice presentation.
Thank you,
Okay,
sir.
I have a few questions actually. Are these qPCR data or protein expression data? What are these?
So these are
You know,
protein expression data.
protein?
Protein expression data of these genes.
Are you sure?
Yes, sir. That is at least what I understood after reading the report.
Please go to your limitation slide. I think one of your limitation was just give me a moment. Okay, actually, this looks like a qPCR data to me. I don't know, it could be protein expression as well. But anyway, so I
I
think,
think, yeah, one of them mentioned as well in the limitation that
yeah, I think
this is
your
not,
partner did mention about the
yeah,
protein
this is
expression
not an
thing.
indicative of protein expression. Yeah.
And okay, that being said, that is one thing. Another thing is BDNF. It's there in one of your slides. What is that?
So it's
Brain-derived neurological
yes,
factor.
neurotic factor.
What is the importance of this in this study? I mean, why are we studying this?
Sir, it is important because BDNF, it promotes neuronal growth, development, and it's widely known to increase it to be increased by
Memory. Yes.
it helps in brain development.
Yeah, and it
Again,
helps also in memory.
cortisol levels. Why are you checking cortisol levels?
Um, because in this experiment, they basically it's not solely based on autophagy. They measured a lot of other things like glucose, cortisol. So they were just trying to see the effect of intermittent fasting on various factors and various components in blood.
And
Okay,
sir,
one of your,
only
yeah,
yes.
please go ahead.
It was only seen, it was only observed that cortisol levels increased or decreased by a significant amount other than other hormones.
Okay, fine. And then you mentioned about SIRT1, SIRT1, right?
Yes, sir.
And it promotes longevity. That was there in one of your discussions, I guess.
Yes, sir.
How?
Sir, sir, it promotes insulin secretion and action and regulates fat metabolism, protects against inflammation, oxidative stress, and DNA damage.
What are sirtuins?
Uh, Sir, I don't actually know. I need to
Anybody from the audience know what is sirtuins?
Sirtuins are NAD plus dependent protein deacetylases or ATP ribosylases, which tend to like chemically modify the various metabolic enzymes which are required or which are the determinants of lifespan.
And how do they do that? Any specific function of sirtuins? Have you studied about it in this lecture? In this entire
A bit. It was, as far as I remember, it was related to the sirtuin 6, which actually causes deacetylation of one particular enzyme.
SOD2, right?
SOD2, right? It causes deacetylation of SOD2, which renders it
Activates
inactive
it.
and which activates it, thus causing like reduction of ROS in the mitochondria.
Okay, so we have actually gone through, why I asked these questions was we had gone through a very similar, well, while we were talking about calorie restrictions, we were also talking about these same genes and also the effect of, the effect of increase of sirtuins and, other autophagy factors on aging. We had discussed about it in detail. That's why I wanted to ask you whether you know about it. But other than that, good presentation, good slide space utilization. Your figures should be greater, larger than your text, as has been pointed out by one of the panelists. Other than that, everything is fine. Thank you for this. It was a good presentation.
Thank you, sir.
Who would present? I think we have only 2 more members. Antik and Shinjon are left, I guess. Who's going to present? Who's going to present first?
So I can.
Okay, please go ahead.
Is it visible, sir?
Yes, yes, yes. Very colorful slide.
Yes, sir. So, good afternoon, sir. I'm Sanjay Sinha, and I will be presenting today on the topic or research article hypersensitivity to contact inhibition. So, nowadays, whenever we hear of cancer, the first thing what comes to our mind is death. Like, why? Because it has no complete cure of it, right? So, but what actually is cancer? It is uncontrolled cell division that result in accumulation of mass of cell, often called tumors. Now, there must be any mechanism to prevent it. So, built in our body has an special anti-cancer mechanism, and we'll be talking about it. So, I have divided my presentation in four parts, where in the beginning I'll talk about the mechanisms of anti-cancer of the anti-cancer mechanism. Then I'll talk about a special rodent, and then up to the experiment, and then what are the observations which we found out. So, mammals have evolved many anti-cancer defense mechanisms involving cell cycle checkpoints, apoptosis, replicating senescence, and which is controlled by tumor suppressor genes as P53 and RB, and contact inhibition. But here I would like to draw your attention on contact inhibition. Contact inhibition is a key anti-cancer mechanism that arrests cell division when it reaches a high density. The cell division stops when the cell comes in contact with each other and thus defines the word contact inhibition. As a result, a normal cells stop proliferating when they form a monolayer. This growth arrest and contact inhibition is signaled by membrane protein and is mediated by elevated levels of P27-KIP1A cyclin dependent kinase inhibitor. P27 binds to the cyclin CDK complex and arrests the cell in G1 phase. In mammals, especially in humans, contact inhibition is triggered by increase in level of P27-KIP1 gene. But when proto-oncogenes are converted into oncogenes due to some actions of carcinogen and one develops cancer, the individual develops low level of expression of this gene, which actually to us seems that cell has lost its property of contact inhibition and leads to poor survival of individual. Now, I would like to draw your attention on our research subject, the naked mole rat. So, a naked mole rat, why is it so special? It is because it has extraordinary resistance to cancer as tumor formation does not occur in this rodent. This is solely because of the special property of contact inhibition. So, in humans, if this property is lost, then what makes this rodent special? That this property is not lost in them. In humans and other organisms, we saw this property was controlled by level P27 Cape1 but this is not the case for naked mole rat. In case of naked mole rat, the induction of P16 Inc4AB and P27 Cape1 are responsible for controlling this property. This property of naked mole rat is that P16 Inc4A and P27 Kip1 are temporarily separated and perform two different functions. Now, moving on to the experiment. So here we have the hypothesis made as naked mole rat do not develop tumor, right? So what we have set up as in an experiment to confirm the naked mole rat do not develop tumor, a normal rat and a naked mole rat were taken, and oncogenic RAS and SV40 large T carcinogens were passed through. The result came quite unexpected to the researchers as the combination was sufficient for the mice to develop tumor in the case of naked mole rat as well. But in case of it, they could hardly see a single layer of cell. The reason uh, given for this was the cell of naked mole rat were hypersensitive to contact inhibition. Coming up to the results which they draw, where oncogenic RAS and SV40 large T do not encourage independent growth of naked mole rat fibroblast. The researchers tested if naked mole rat fibroblast could be transformed into cancer cell using combination of these carcinogens, which transformed mouse cell. Naked mole rat cells are unique because they have the endogenous telomerase activity, rendering them immortal in culture. SV40LT comes in various forms, Wild-type LT inactivates both P53 and PRB tumor-suppressing pathways. LTK1 inactivates only P53 and LT delta 434, 444 inactivates only PRB and related proteins. When mouse skin fibroblasts were infected with oncogenic RAS and SV40LT, so its mutant form, they formed robust colonies. In contrast, naked mole rat fibroblasts remain single or formed small incomplete colonies. This suggested that naked mole rat acquire more genetic change or hits than mouse to become cancerous. In summary, the demonstration that naked mole rat fibroblasts display a significant resistance to malignant transformation when exposed to oncogenic RAS and SV40LT, highlighting their unique anti-cancer qualities. The second observation or result was naked mole rat cells are hypersensitive to contact inhibition, as we have discussed before. The growth of naked mole rat fibroblasts in vitro were examined. Mouse as well as human fibroblasts are typically cultured by seeding 5 into 10 to the power 5 cells per 100 millimeter dish, as you can see here. When we attempted and then splitting the cell when they reach confluence by forming dense monolayer. When we attempted culturing naked mole rat cells under the same regime, we could not obtain any cell proliferation. A solution to the problem was found that we plate naked mole rat cell at lower cell density, 5 into 10 to the power 5 cell per 100 millimeter dish. When plated at low cell density, naked mole rat became proliferate, but because confluence upon the formation of first cell contacts. This is in sharp contrast to the mouse fibroblast that forms dense monolayer of cell at confluence. The third observation was early contact inhibition is associated with induction of P16. Naked mole rats cells exhibit a unique two-tiered contact inhibition mechanism, which is not seen in humans or mice. Early contact inhibition in this cell is associated with accumulation of P16, while regular contact inhibition is linked to P27. Confluent naked mole rat cells show minimal P27 induction, but if contact inhibition is perpetuated, P27 level increases. The loss of early contact inhibition also makes this cell prone to encourage independent growth. Importantly, P16 induction is rapid in response to cell contact, and PRB becomes hypophosphorylated, consisting of P16 activation. This two-tiered contact inhibition system likely enhances naked mole rat resistant to cancer, setting them apart from human and mice. Summarizing, these kinds of results were due to the fact that P16 Inc. 4A and P27 KIP1 are separated and target two different functions of the contact inhibition. The enzymes P16 Inc. 4A control the early contact inhibition, while P27 KIP1 is not induced in cell undergoing early contact inhibition, but accumulates in transform cells that have lost contact inhibition and arrest at high cell density. Thus, both act complementary to each other, and also early contact inhibition is dominant over late contact inhibition, which basically does not allow the level of P27 KIP1 level fall, and since if level of P27 KIP1 doesn't fall, then there is no chance of losing the property of contact inhibition. Thus, in naked mole rat, it has two level of security against cancer, which is not so in humans, rat, or any other mammals, which makes this our special rodent so special. Thank you. I have provided the DOI as well here, and I am open for questions.
Okay, Shinjan, so many things to tell you, but I would ask someone from the panelist or the audience to ask first. Agrideep, maybe.
It was a nice presentation, Srinjan. I liked it. And yeah, no technical questions. I'm still processing. But yeah, It was a nice presentation.
Orko?
Thank you, sir.
Yeah, so I just was thinking, like maybe like if you said like what is the conclusion? Like early contact inhibition is better for preventing cancer, right?
Yes, sir.
Is that
Until
the
and
takeaway?
unless the level of
Okay,
P27 KIP1 doesn't fall, the early
okay.
and the P16 level rises, the cell will undergo early contact inhibition, and thus it will become the first barrier, and cell won't lose this property.
So would you also say that that property might, aid or promote senescence in cells
No, sir.
because there
Here's
is
the catch.
early,
Yes, sir.
yeah, yeah, go ahead.
Here's the catch that this what would you say, this targeting this P16 is only triggered when it undergoes under some carcinogens, right? We have seen
Okay,
this rise in P16 level because it has undergone this RAS oncogenes, right? If it wasn't given that much concentration, this P16 level wouldn't hyped up. In general cases in naked mole rats, the level of P16 is not always that high, but when oncogenes were passed through it, it showed the activity, so it won't affect in the senescence of the cell.
right. Okay, okay. And regarding the presentation, it was a very nice presentation. Nothing to say. So, well done.
Thank you.
Yeah, Shinjan, if I understand correctly, there are two levels of contact inhibition in naked mole rats, if I'm not wrong.
Yes, sir.
Okay, so that is better in than the normal process,
Yes, sir.
right?
Yes, sir.
Okay, okay. Apart from that, one of your slides showed RAS plus a deletion of 434. Maybe can you go back to that slide?
Yes, sir. One second.
Yeah, that's the next
Yes,
one.
sir. Yes, sir. This one.
Yeah, RAS, the third panel, the RAS plus, it's a delta 434. What is it?
Yes, sir. So it's a mixture of oncogenes, sir.
Okay, okay, okay. So the
So this
conclusion,
delta 434, as per as I understood, it actually helped in heating as well. Like the cell got heated up.
heated up.
Yes, sir. It was a small section written there, like which was to understand that I googled it for the summary as well. Like what is the function of each of them? So there I that what I inferred from it.
Okay. Okay. Anyway, I would say it is a good presentation. Your presentation skills are good. Slides have been structured well. The results have been spread out properly for every one of us to understand. One thing was maybe a use of pointer would have been better because you were talking about a lot of things and we were not exactly following the exact panel which you were talking about, but it's fine.
Yes,
Apart
sir.
from that, what
So I don't
else?
know where can I get pointers. I tried a lot, being
Can you
honest.
right click right now over here?
Yes, sir. I'm doing it.
Nothing is coming up.
No, sir.
It should be there in the right in the below bottom left corner if you see.
For this one. Oh shit.
Yeah. So
My bad.
yeah, you could use it. It is better. Apart from that, really, really nice presentation. Very well, very less text also. So yeah, thank you very much for that. It was a very good presentation.
One suggestion from my side, Srinjan, if you go to slide 8, maybe I forgot the middle result slide.
Yes,
The
sir.
one,
This one.
this one and the slide 7. Can you go once?
Yes, sir.
Yeah, and whenever you are explaining slides like these, use your pointer to exactly point out what you are explaining. As Sanket already said that let's say this is GFP plus LT, and you like explain the audience that you see a very less cell density, but when these are the RAS, these are the oncogens, and then this is the result. So that audience can actively see what you are telling. So that would have been much just an improvement, but
Sure,
otherwise,
sir.
yes, right.
But
So
it
that
Yeah,
time I
appeared that you
didn't
were
knew
very
where.
thorough with your presentation anyway,
yeah,
so it is a good thing.
So I actually didn't know how to use this pointer, like from where I
yeah.
could get it.
But
Next time I
it's a
would keep it in mind.
yeah.
Thank you so much, Shinjan. Anybody
Thank you, sir.
else? I think Shagnik and
Yes, sir. Sir, our team is there.
okay, okay, please.
Okay, sir. Presenting. Sir, is it visible?
Yes, yes.
Hello, sir. Am I audible?
Yes, yes, both of you are audible.
Can I start?
Yeah, sure.
Hello, good afternoon, everyone. I am Sharmi Chakraborty and my teammate Aditya Mujumdar. Today, we're going to represent a presentation about the extension of murin lifespan by the overexpression of catalyst targeted to the mitochondria. Their murin represent the mice, and that is important in topic about the aging process. Next. Uh, there, in the introduction part, the ROS, the reactive oxygen species, a important part in the radical theory of aging, propose that the ROS in the biological system attacks the molecules and cause a functional decline of organ system that eventually leads to death. And very important that in the ROS generated mainly from the single electron escaping of the mitochondrial respiratory chain and reducing molecular oxygen to form a superoxide ion. That's SO2 that's converted into the H2O2 and which produce a highly reactive hydroxyl radical. Also, we see that from this right side picture of the slide. Next. Uh, in the experimental part of this research, that we have to determine uh, the role of the reactive oxygen species of mammalian longevity, which generated transgenic mice that overexpress the human catalyst that localize the PCAT, MCAT, and NCAT respectively. In the also we see that transgenic mice supports the free radical aging theory or not. That also we represent. In the we see that there is a right side graph. There, the four graph ABCD. That AB that represent the catalyst activity in the heart and muscles, and the B and the C only in the one slide are increasing that from the brain, and the D graph also that in the mitochondria fraction amount of catalyst increase 50%, that more than the of the wild type. In the observation part, we see that in the right side picture that there A and B represent that the cardiac tissues, and the C and D that's at the embryonic fibroblast cultures. And in also we observe that the MCAT with detectable level of human catalyst. Also, that the catalyst that the PCAT animals showed that the limiters until the death and the 3.3 months in the first organism and the second organism in the 3.5 months, and the MCAT for the 1 month and the second organism the 3 months, and MCAT for the 4.5 months and the 5.5 months from the second organism. Also, in the graph figure, in the further slide that my another teammate Aditya Majumdar represents for.
Sir, mm, as explained by my previous teammate, in the graph, in this graph we can see the overexpansion of the lifespan, median lifespan in all the three cases. Then we perform another test, which is the graph number D. Here we breed hemizygous PCAT overexpressed animal with the SOD with the animal with have overexpressed SOD1 gene. This double transgenic mice has actually 18.5% extension of median life compared with wild, and 7% extension with compared to the PCAT ones, as you can clearly see in this 4D graph. So we can from this we can conclude that this combination of SOD1 and SOD2 with MCAT can even cause greater extension of lifespan longevity in mice models. Then in this experiment we measure the aconitis activity. Actually, when ROS attacks mitochondrial tissue, it damages the enzyme. But in case of overexpression of catalase, it can break down ROS, and aconitis activity is largely increased. As you can see in this figure A and figure B. Here another experiment, we treat the mitochondrial tissue with H2O2 also. So in case of H2O2, we see that amount of aconitis is to some extent less in case of wild type, but in case of MCAT overexpression, the aconitis activity is as it is normal, as H2O2 is damaged by H2O2 and other ROS is removed by catalase. So when we see that measure the activity of aconitis as a ratio of before and after, we see it is threefold in case of 5 month and 43-fold in 19 month and 950-fold in case of 30 to 33 month old mice. In the next graph, in the 4D, we can see that the H2O2 production is also reduced. As you can see this 4D graph, in case of MCAT, the amount of internal H2O2 is less than the wild type. Then another test, we see that in case of overexpression, whether any oxidative damage occurred to DNA or not. There is a we can measure the 8-hydroxydeoxyguanosine when isolated from genomic DNA analysis of skeletal muscle and heart. We see in all the types sample the age-related increase in case of 8-OHDG, but in case of MCAT, the increment is to some extent less, as this is protected from ROS activity. Next, in the figure 4F, in the figure F, we observe the mitochondrial deletions associated with the oxidative damage. Here we can isolate and see various low molecular weight marker and quantify their number. In case this, we observe this increased with age in case of both wild type heart and skeletal muscle, but same in case of previous, it is much less amount of oxidative damage product in MCAT skeletal muscle is observed with compared to the previous one. We also then measure the cardiac histopathology conditions of the mice models. Young mice and older mice were taken. This cardiac measurement of cardiac histopathology is one of the most important feature to, and it is associated with the aging process. We take the severity of the heart pathology on the scale of 0 to 4. In case of both MCAT and wild type mice, two heart pathology conditions are observed. One is arteriosclerosis and another is cardiomyopathy. In both the cases, severity of expression is less in MCAT than the wild type, as you can see in the table. Thus, the table demonstrates the potential of the MCAT protein to protect the heart, and thus the mice have a prolonged health span as well as the lifespan. Then in the last part, we see we also measure the cataract, the amount of cataract falling in the eye. In MCAT overexpressed mice, we see that amount of cataract is much less than in case of wild type. But when the mice becomes aged gradually, at 27th month, the trend is a borderline significance. That means more or less similar amount of cataracts are formed in both the types. Then we come what we interpret to the experiment in the research. In the research, we have found that MCAT longevity phenotype is there is a longevity in MCAT phenotype, but it occurred both from the direct effect of oxidative stress and also from the indirect secondary intracellular H2O2 dependent signaling, which indirectly also plays a role in the longevity. There is another interpretation. There is mosaic pattern of catalase. This mosaic pattern actually occurs from the selection against cell which express high catalase. This selection because ROS can be an important mitogen. Then when we perform the experiment, this transgenic experiment after taking the organism continuous hybrid embryo backcrossed with the founder pure line organism, that means after nearly 7 to 8 generation, we see that the MCAT protection against the mitochondrial toxicity, DNA damage, and deletion is to some extent less than what we see in case of mice, transgenic mice taken 2 to 3 generation after cross. Thus, we can clearly support that our experiment supports the free radical theory of aging by lifespan extension in case of mice model. In the experiment, we have assumed that the aging, median lifespan, aging, and cardiac pathology experiments are performed nearly 2 to 4 generations after generating the transgenic organism. But entire biochemical assays like aconitis assays, such assays was performed after generation 8 or 9, and the mice has been moved to a facility where life extension and phenotype is diminished. So I can clearly conclude that this mitochondrial ROS is a very important limiting factor in determining this mammalian longevity, and it can provide further impetus to study on new and combined antioxidant mouse models in future. In future, a lot of experiments aggregated from this mice model. Thank you, sir, for your guidance in entire course and for presenting the paper. I would also thank our college. Thank you, sir. Now, team is open to questions.
Yeah. I think we will ask the panelists first. I'm getting a lot of acknowledgements. I'm not sure whether they are honest or not, but anyway. Let's go to Agrideep, maybe.
Hi, I have a simple question. I don't know. Can you go to your introduction slide?
Yes.
Yes. Here in this diagram, you have said that plasma membrane contributes to ROS. Do you have any examples? I mean, I don't know if there are any, but this is the first time I saw plasma membrane contributing to ROS. Can you, I mean, yeah, are there any examples or like anything else from the paper that you came across? Or was it just a diagram?
No, sir.
Is this a diagram from the paper that you?
No. It is actually it is. I have tried to depict how ROS is generated, but I have get this image from not from paper, out of sight.
Okay, and in that case, whenever you are, I mean, it's just a suggestion because yeah, I understand that many of the images that you get from Google, you have to first properly attribute them, that where they're from, and second, if there's something in the image, you will be open to questions for that particular thing and be prepared for it. And next is, I would say you can like improve your slides by a lot. There are certain slides like slide number 5 or slide number 3. They're like the let's say if you go to slide number 5, the the color and the yellow text, this is like it doesn't look particularly good if you are presenting in front of a large audience. We are here just to like give you suggestions, so let's say, so that you can improve later when you are giving your final presentations or anything. But yeah, keep that in mind to always choose lighter colors and avoid yellow texts. It's like a warning sign in front of a highway. So
So shall we give dark colors?
take a lighter background and use
Yes.
black, dark brown, dark blue, anything which is not very flashy. Your presentation needs to be sweet. It's not like you have to warn people that, hey, this is the observation. This is the observation. Catalase in MCAT samples is 50% higher. It should be soothing to the audience. It should like fit the story that you are telling. Okay, and then uh, uh, yeah, a similar few like suggestions for the sixth, seventh, and eighth slide. If you go there, you keep I would suggest keep the figure in the middle, but as you have a column, a figure column, and this would be particularly difficult. So you can just crop the graphs and just make multiple slides. But as your graph is just a single particular graph, so it won't take much time. So you would be like making, let's say, 6 extra slides, but the content that you are telling is similar, so it won't take much time. There's no limitation on the number of slides. It's just you are just limited to the amount of time that you're given. And yeah, again, avoid the yellow texts, and yeah, give page numbers. And these are the like the
So
common
that
suggestions.
means we would give one graph in each slide.
Yeah, that's much better. That's much better. But I don't know if I have a paper that I presented or not. Uh, Sanket, I don't know if you have time
Yeah,
here.
maybe at the end we can do something
Yeah,
regarding that.
yeah. At the end, if I find it out, I will just show you one of the slides that I presented here.
Yes.
Maybe it will help any of you. Okay, but otherwise, yeah, nice try, and keep going, improve your slides, work on the suggestions. Thank you.
Yes. Thank you, sir.
Orko?
Yeah, I think the suggestions have all been given. So, can you go to slide number 4? Yeah, just let me. So figure A is from
Figure
heart
A is
and
from
figure B is from skeletal muscle, right?
no, sir. Two organisms. A denotes one organism and B denotes another organism. Two founder line organism, and suppose in one particular organism we have found
Okay,
the catalyst
okay.
liver, kidney, heart, and muscles. Two organisms.
Right. So, so yeah, I just in this slide only like I was confused. I just wanted to mention like write a little more better in a better manner, and you can just like
Yes.
enlarge the slide that graph
Yeah.
as well. You can put maybe two graphs in one slide. One, if not one, maybe two, not four. So that was another, and I think rest it's all been addressed. So yeah, okay.
Okay, from my side, of course, the color scheme has been a little bit problematic. Did you guys do this separate parts of the slide separately or something like that? Because till slide 4, there is one pattern, and then it appears that the scheme has totally changed from slide 5 to the rest. So,
So
I
what
mean,
you
do
do
you
scheme?
the theme, if you see a
Theme.
presentation should follow a uniform theme throughout, right from the beginning till the end. So that is not exactly here. If you see, apart from the other suggestions, which are of course true by the other panelists, this is one another thing I would like to add up. Another thing is what are PCAT, MCAT, and NCAT?
Is this PCAT actually we generated transgenic organisms, three types of transgenic organisms. In one type we overexpressed this nuclear catalase, and another in mitochondrial catalase, and another in peroxisomal catalase. 3 types of transgenic biases.
That answers the question. Yes. Okay. Good. Another thing is you said aconitis activity is lowered or it is increased when you have higher catalase activity, right? Somewhere you have mentioned this.
No, yeah, actually, when there is a higher amount of ROS, then aconitis activity is low. And
why so?
Because ROS damages mitochondrial tissues by various mechanisms, and one of the mitochondrial enzyme is aconitis, so aconitis activity is much reduced by ROS activity.
There are many other mitochondrial enzymes. Why do you choose aconitis? I mean, why did they choose aconitis? Do you remember anything about ROS and aconitis? Any reactions that we have studied? What are the enzymes that convert hydrogen peroxide to water and oxygen? There are two enzymes. One is catalase, another one is
That is the SOD.
superoxide dismutase. And
Yes,
what if
superoxide
this
dismutase.
hydrogen peroxide is not converted? What is the other reaction that happens?
Can as question once more?
What happens if hydrogen peroxide is not converted to water? It is not neutralized. What would happen? Do you remember Fenton reaction?
Yes, sir.
Have you heard about it?
Yes, sir. It is.
It is?
So it is actually one kind of biochemical estimation test.
No. I think you're confusing it with something else. We have it in one of our slides about this mitochondrial theory. I mean, mitochondrial ROS production. There we have that the 4Fe4S clusters of
Sir, the
enzymes.
reaction in which OH hydroxyl radical is generated from H2O2.
Yes, hydroxyl radical, which is more damaging, and it interferes with the 4Fe4S clusters of enzymes, which is one of them is aconitis. Do you remember something, or you have forgotten?
Sir, no, sir. Actually,
Okay,
remembering
I
to
think
some extent.
yeah, just go back through the lecture slides. I think you will remember it. Another question: free radical theory of aging. What is it? You have mentioned it. This study supports free radical theory.
Actually, this theory which depicts that various free radicals like ROS processes aggravate this aging process. This is actually free radical theory. That means a theory in which ROS takes part in the damaging biological molecules which aggravating the aging procedure.
Okay. These are just some technical questions to you. If you present somewhere, maybe you'll get questions like this. But anyway,
Yes.
good presentation, nice work, and it really aligns with our course. At least I could tell you, really nice. But please brush up some of the concepts. We are going to have the MCQ exams next week. So yeah.
Please, I'll be looking after the slides. Your slide.
Okay. Thank you.
Thank you,
Anybody is left?
Sir, I am left. Hello, sir.
Yes, please go ahead.
Okay, wait. Screen window. Okay, wait, sir. Sir, can you see the screen?
Yes, it is visible.
Okay. So, good afternoon, everyone. My name is Antik, and today I will be presenting on the topic of the SIR234 complex and SIR2 alone. These two gene complexes, they promote longevity in Saccharomyces cerevisiae by two different mechanisms. So now, first we would like to understand that what is basically SI or a SIR, which I'm referring to. It is basically silent information regulator, and these are the most critical genes which are the determinants for lifespan in yeast mother cells. Now, there are four classes of these silent information regulator genes: 1, 2, 3, and 4. Now, these genes, they code for certain sirtuin-like proteins, which basically perform the similar function as sirtuins, but these are not exactly sirtuins, but they share a much similar homology as compared to the human sirtuin proteins found in humans. So these are also some NAD plus dependent protein deacetylases or ADP ribosylase, and their functions and some of their important functions are non-homologous end joining. These play a specific role in non-homologous end joining, and they also play a role in transcriptional repression of the silent mating type loci of HML and HMR, which are basically HML is responsible for expression of the alpha mating type of the yeast fungus, and the HMR is responsible for mating of the A for production of the A mating type. Now, why this transcriptional repression is required? Because it is used to prevent haploid sterility. Because if these third genes are not present, then haploid sterility, then it would result in haploid sterility and cause a shorter lifespan of the yeast cell. This would also cause a positional effect variation of gene expression, which mainly occurs at the telomeres, for which the telomere length, for which it also maintains the length of the telomeres. So now, what was the aim of this particular research was that we would like to understand the mechanism which underlies the lifespan elongation in Saccharomyces cerevisiae by SIR234 complex and SIR2 alone. So we will just go to the results. Like very simple experiments were done. So first, what was done that W303 MAT A RAD5 this strain of yeast was used, and then three individual sets of yeast cells were made. One was a SIR2 knockout, one was a SIR3 knockout, and another one was a SIR4 knockout. Now, it was observed that SIR3 and SIR4 knockouts, they had a 20% lifespan decrease, whereas a SIR2 mutant had a 50% lifespan decrease. So it was already considered of the fact that SIR2 had a much had a much predominating effect on the lifespan of the yeast as compared to SIR3 and SIR4, but also SIR3 and SIR4 had some very distinct effects on lifespan, which we will see in the later slides. So uh, two genes, HDF1 and HDF2, these genes were also knocked out in some in the SIR2 and SIR3, SIR4 mutants subsequently, and this was it was also seen that subsequent lifespan decrease was there. But so initially it was found that since HDF1 and HDF2 genes are responsible for non-homologous end joining as depicted in this picture, so might be non-homologous end joining would have been a reason for, like, probable reason or underlying mechanism for this reduction in lifespan. But then it was also shown that HDF1 and HDF2 are also crucial in telomere maintenance. So there was a pleiotropic effect which was observed in this case, for which the scientists, what they did is that they mutated, like they knocked out the yeast DNA ligase 4 gene, which is essential in the last step of non-homologous end joining, that it facilitates nick translation. But still it was seen that non-homologous end joining did not cause but still it was seen that the DNL4 minus, which is the gene for yeast DNA ligase 4, this knockout did not cause any reduction of lifespan. Hence non-homologous end joining was understood to be not a cause. And this was also observed in these two. I guess you remember, we will be able to see this. So it was also observed in these two that the SIR2 mutants, they had a much decreased lifespan. These cross signs, these represent the SIR2 mutants lifespan, and the other ones SIR2, 3, and 4, SIR3 and 4 respectively. And there is also a wild type which is represented by this by this square-like denotation, which indicates that SIR4 and SIR3 might have a reduction effect, but the effect of SERT2 is much greater. So now we come to the second observation, the second result, that this short lifespan of the SERT3 and SERT4 mutants, as was given in this, as was said in this slide, this was due to the co-expression of the A and alpha mating type genes. Now, what is this A and alpha mating type genes? Let me explain a bit. The thing is that any fungus, in general, any fungus, it always remains sexually functional in the haploid condition rather than in the diploid condition, for which its lifespan because, and that is why it has a haploid like haploid dominant life cycle as compared to other organisms which usually have a diploid dominant life cycle. So now these SERT2, SERT3, and SERT4 genes, these they do what they do. They transcriptionally repress these HML and HMR alleles, for which the A and alpha mating type genes do not occur. Now, if I knock out any of these genes, this will cause a simultaneous expression of this A and alpha mating phenotype. But this could be subsequently neutralized by deleting any one of the alleles, that is HML or HMR. If we delete HML, the alpha mating type gets deleted, for which in the null SERT3 and SERT4 yeast, when we are deleting HML alpha, the lifespan is extending. Whereas in only the sole SERT3, SERT4 knockout strains, there is no expression, but in the no extension. But SERT2, HML alpha, and SERT2KO had a similar lifespan, which indicates that deletion of HML alpha in case of SERT3 and SERT4 is more important as compared to deletion of the same allele in SERT2KO. That means SERT2 knockout, that means SERT2 gene is not a very crucial gene in transcriptional repression of these HML and HMR locus. But this depression of HMR loci, they caused haploid sterility, resulting in a shorter lifespan. Now, another subsequently mice was taken, and in its URA3 locus, MAT alpha gene was integrated, for which SERT4 HML and SERT3 HML strains showed a reduced lifespan, because SERT4 HML and SERT3 HML means the SERT4 and HML, and here SERT3 and HML strains were deleted, for which lifespan was extending, but resulting in the A mating type strain. But we are additionally adding the mating type alpha strain, which causing again haploid sterility. So these results were again subsequently understood by another experiment, that homozygous SERT2 diploid mutant diploid, they had a shorter lifespan than this wild type diploid, whereas mutant SERT4 had no difference with isogenic with a similar strain having an isogenic character of alpha. Now, so therefore, these SERT4 and SERT3 knockout has a negative effect on the lifespan by transcriptional repression of silent mating type loci, HML and HMR. So this is one particular mechanism with the help of which we can understand that what is basically happening. This is also understood by this particular graph, where the lifespan is getting decreased. Now, with respect to SERT2, the lifespan with the wild type is not much varying. Now, the next one is that is short lifespan of SERT2, SERT3, and SERT4 haploids. They are likely due to ERC formation. So, in the yeast cells which had been knocked out, the three basic things were observed. First is NHEJ mechanism. The second one is A and alpha mating type gene, and the third one is the extrachromosomal rDNA circle or ER. Now, what is ERC? Now, how these ERCs are basically forming? So, in the nucleus, as we know that in humans, the sirtuin proteins are localized within the nucleus, the crucial sirtuin proteins. Similarly, in yeast, the SIR proteins are also localized within the nucleus. So, what these basically do, that they also play a role in during the replication and transcription of the ribosomal DNA of the 35. So basically, the ribosomal DNA is. Let me just draw it. It's just a very rough diagram. So if we have this ribosomal DNA, we have this 35S here. And we have this 5S coding gene here, and in this place we have an NTS or non-transcribed spacer, which actually contains one protein, which actually contains one sequence known as RFB or replication fork barrier, to which one protein known as FOB1 protein binds, for which the replication of this ribosomal DNA is unidirectional. Now, since this the replication is unidirectional, it causes breakage of the DNA. triggering homologous recombination. Now, due to this homologous recombination, the ribosomal DNA often forms circles. So, but these circles are actually so it has been seen that an accumulation of ERCs in yeast causes decreased lifespan. So now now here comes the main role of SIR2, that SIR2, it basically represses this homologous recombination and hence increases the lifespan. Now the SIR2 knockout in yeast, that they showed increased levels of ERC formation and hence a shortened lifespan, as it is already said. But the main question that was that is SIR3 and SIR4 also responsible for this? It was shown that the reduction of lifespan in case of SIR3 and SIR4 mutants was not very significant. But there was one significant difference, that ADE2 marker gene, which was actually cloned into the SIR3 and SIR4 mutant yeast. So it was seen that it was lost at a 2 to 3-fold higher frequency than the wild type. So in the SIR3 and SIR4 mutants as well, there is a decrease in lifespan and increased ERC accumulation due to loss of this ADE2 marker gene and subsequent loss of this ADE2 marker gene, but it was not as significant as compared to the SIR2 protein. So therefore, it was understood that the result was not NHEJ. The first result was A and alpha mating type genes, and the second one means A and alpha mating type gene co-expression, for which the lifespan is reducing, and the other one was also the ERC formation. And this release of ERC in the wild type nucleus, what it causes, it causes a shorter lifespan in the wild type, and also in the like in the natural strains like SIR4 plus and SIR3 plus cells. If we isolate one extra chromosomal RDNA circle and insert in these, that also causes causes a reduction in lifespan to some extent, but not it is not such a very dominant effect, but it actually occurs. And next we have the deletion of FOB1 gene. So FOB1 gene actually it causes release of FOB1 protein, which reduces, which actually causes DNA hyper recombination and lifespan and a decrease of this of SIR2 mutants. Now, what basically happens is that we will focus in this agarose in this autoradiography film. So what the experiment was done in the young and old type cells, the wild type SIR2 delta and HML delta, SIR2 delta, HML delta, FOB1 delta was processed and their DNA was isolated. Similarly, it was done in the old cells. Now, these single arrowheads, they represent the chromosomal DNA, and the old cells, they represent the, and then these arrows, they represent the formation of ERCs, small and big sized ERCs, that depends on the time. But interestingly, these results, they do not actually match the inference, is that we can see that there is very little ERC accumulation here. But in the old, whenever we are deleting the SIR2 delta and HML delta, it is causing a huge accumulation of ERCs. Now, the thing was that this FOB1 gene, it causes, now how was this effect reversed? If we just look carefully, that SIR2 delta and HML delta was, when it was done, it caused ERC accumulation. But when simultaneously FOB1 delta was deleted, this same band actually vanished. So what was the reason for this? The reason for this is that since we know that FOB1 gene produces an FOB1 protein, which actually binds to this non-transcribed space, as I had mentioned earlier, there is a replication fork barrier sequence to which the FOB1 protein binds, for which the DNA polymerase cannot travel in one, but in this direction. Rather, it has to travel in one direction only. Now, if we delete this gene, it activates the bidirectional form of replication, which causes reduction of rDNA homologous recombination. Because since we are dealing with bidirectional replication, so the frequency of DNA damage and DNA break, but double-stranded break, decreases, and for which the lifespan increases by 30 to 60%. And also, it was seen that SIR2 HML strains, they also showed a significant decrease in the lifespan as FOB1 was knocked out, but it was less than sole FOB1 knockout in yeast, which is very natural because of the fact that if we only delete FOB1 knockout yeast, then it might be said that the frequency or the probability of DSP occurrence is zero, basically, because DSP can only occur when FOB1 gene is present, when FOB1 protein is present. So if we are deleting that, the source of this FOB1 protein, so the rate of recombination decreases. The second result, the last result of this research was that the SERT2 dosage increase, it decreases the lifespan. Interestingly, it was seen. Interestingly, it was seen that heterozygous SERT2 means like dominant SERT2 and recessive SERT2. The small SIR2, it indicates the homozygous. It represents the null allele, and SERT2 represents the dominant functional allele. So this had a lesser lifespan. Means whenever we are deleting one SERT2 gene, this causes a reduction in lifespan, which obviously infers the fact that, but it never experienced instant fatality. But on the other hand, when it was cloned with another, when this yeast was cloned with another SR2 gene, the lifespan increased by 30%, which followed the fact that that the inheritance of SR2 genes does not really follow Mendelian genetics; it follows non-Mendelian genetics more. And this SERT2P, it actually causes a reduction. This actually causes since the SERT2P is the protein produced from SERT2, which actually causes decreased ERC accumulation, and hence it is a limiting factor for the lifespan determination in these mother cells. We will also see that it has prevents functions in two stages, for which it is a determining lifespan. And not only that, the SERT2 does not prevent recombination of the blocked replication forks, but it prevents homologous recombination, hence ERC formation, as is evident from a similar lifespan as only FOB1 strain upon additional SERT2 cloning, which was basically mentioned previously. But FOB1 delta is only responsible for binding to this non-transcribed spacer, and SERT2 is only responsible for reducing ERC accumulation. It does not block ERC accumulation. If you want to block ERC accumulation, then we have to produce an FOB1 delta strain. And if we have to only see the prevention and hence extension of lifespan, then we have to delete only SERT2 gene. So it is also shown in this two graphs as well. So SERT4P, it does not function to extend the lifespan after less of an ERC. SERT4P is not so much important, but this mutation of FOB1, it suppresses the lifespan defect. So we can see the lifespan, it is decreasing to a huge extent as compared to the only sole SERT2, SERT3, or SERT4 mutants. I told the wrong thing. Sorry, the mutation of FOB1 protein, it increases the lifespan. I couldn't follow. Sorry. So the mutation of FOB1 protein, it basically increases the lifespan. So we can see the slope of decrease gets lower, for which the lifespan gets extended. But in the case of only FOB1, we can see that since FOB1 is active, so it causes a drastic reduction in lifespan in case of the SERT2 mutant. Next, and we can also understand one important thing regarding this, that SERT2 but not SERT4 is actually required for longevity in the diploid cells. If we are considering the diploid cell, if longevity is to be considered to be an important factor in the diploid cell, then SERT2 is the limiting mutant. And in case of haploid, we can see that A and alpha mating type genes, the SERT3 and SERT4 genes are mainly crucial. So therefore, all of these results I have tried to interpret in this discussion panel, which actually infers the fact that the SERT234 complex, they extend lifespan in haploid strains by transcriptional repression of HML and HMR alleles of the mating type genes, with respect to which SERT3 and SERT4 genes are most dominant. And this co-expression also causes shorter lifespan, as it was previously mentioned. But this problem could be evaded by deleting HML locus, which induce the fertility in the haploid strain. So we can see, and we can also see that the SERT2P is a limiting component of aging in yeast, which causes lifespan extension beyond wild type upon overexpression. So we can also see that it inhibits the replication fork blocked by this FOB1 protein. So what basically does is, I am trying to again depict it by a diagram. I guess this background is easy. So here we have one sequence known as RFB. So here this FOB1 protein binds. My handwriting is very bad. Pardon it. So this FOB1 protein binds. So what this SERT2P does, it goes and it inhibits the FOB1 by binding with RFB, for which the double-stranded DNA break also reduces to a huge extent. And even if some of the DNA double-strand break reduces, it reduces homologous recombination, thereby promoting non-homologous end joining more as compared to homologous recombination, because homologous recombination is, although it is rare, but inside the nucleus it occurs. But if we totally but SERT2B totally removes the possibility of HR or homologous recombination, for which NHEJ occurs. So this was all about the research paper. So therefore it was concluded that, sorry, there's a pointer. So it was concluded that SERT234 complex and SERT2 alone, they promote Saccharomyces cerevisiae longevity, one by reducing the ERC accumulation with the help of the SERT2 gene, and the other one by prevention of A and alpha mating type information co-expression, which is assisted by SERT3 and SERT4 genes. So this was all about the presentation. I would like to thank my Shanket sir for providing me this opportunity, and also I would like to thank my department for giving this value-added course, so we could learn something out of the box and out of the syllabus for the knowledge. And so thank you everybody for listening to my presentation, and I'm open to questions now.
Yeah, anybody from the panelists would like to go first? I have a lot of things to say. Agrideep?
No technical questions. I liked your presentation, Antik. It was nice.
Thank you, sir.
And it was a big paper, and you handled it well. I saw that
It was actually a very big paper, so I had to study a lot. Otherwise, it was not possible for me. Yeah.
And yes, you have done it quite good.
Yes, sir.
And
thank you, sir.
Yeah, keep it up.
Orko?
Yeah, same from my side. It was a very good presentation. I like the fact that you divided the results into multiple slides, and yeah, it was a very good presentation. And the slides and the color combinations were also nice. So yeah.
Yes, sir.
Good.
Thank you, sir. Thank you, sir.
Okay, Antik, while you speak, I mean, this is a very small suggestion, but while you speak, things get, I mean, you need to speak maybe a little bit slowly. I know you're in a time constraint, but
Yes, sir.
few words
I was thinking that I was trying to slow down, but then whenever I was trying to slow down, I suddenly remembered, oh, I have only 6 minutes time. I have to complete so much. I have to cover up so much. So yeah, it was a time constraint was a thing. Yes, sir.
Some use of joining words, but and only where too frequent. Again, not. I mean, it is after a point, it is going to distract the audience in a way. So that's a suggestion. Another
Sir,
thing is
which phrases, sir, which phrases did I use more?
like you said, but a lot of time. So, but this is happening, but that is happening. So,
okay,
I mean,
okay.
joining words, try to keep it as less as possible. Otherwise, it becomes a little bit annoying to the audience. But it's fine. It's a very
Okay.
small suggestion on that. And another thing
sir.
is, you said homologous recombination is rare. Is it?
I would not say. See, the thing is that it has not been clearly mentioned in the paper that yes, homologous recombination is too much rare. But yes, it occurs. It also occurs, but the extent to which non-homologous, sorry. But the extent to which non-homologous end joining occurs is a bit greater. It is just that.
And how would that be different if we go for non-homologous end joining? Both are DNA repair pathways. If we go for a non-homologous end joining instead of homologous recombination,
Now,
will there be some problem?
The thing is that since homologous recombination, it mainly causes C. But the thing is that non-homologous end joining, it doesn't cause the ribosomal DNA to circularize, but it has been seen. But if we go for homologous recombination causes circularization of the ribosomal DNA, for which the transcription factors cannot accurately bind. This transcription factor part is totally my interpretation. So the transcription factors might not be able to bind as efficiently as it in the linear form. So for which the like the ribosome, like the ribosomal complexes, the amount of ribosomal complexes will be less, for which it will cause a generalized reduction in the lifespan in the metabolism of the yeast cell. This might be one thing.
Okay, you have one thing, no lifespan reduction in DNL4 knockout mice in this very slide, hence NHEGA is not a cause. Could you please explain that?
The thing was that it is just means it is basically an observation. Wait, let me use the pointer laser pointer. Okay, it was just an observation that was made by the scientists that when they did a SERT2 mutant, they observed a 50% lifespan decrease. Then they did what, they knocked out the HDF1 and HDF2 genes, which also caused a reduction in lifespan. But since HDF1 and HDF2 were also like essential in telomere length maintenance, so it was not absolutely clear that whether HDF1 and HDF2 is actually causing a reduction in whether HDF1 and HDF2 are actually crucial in means like whether non-homologous enjoining is actually a crucial cause. So now they studied non-homologous enjoining, and they saw that like this is DNA ligase 4, this which is represented by this XRCC4. So these XRCC4 and XLF. Sorry, the ligase 4 is given. Sorry, these are two separate things. I have juggled up. Sorry. So this ligase 4, what it does, it basically facilitates this nick translation. So what basically happens is that now they knocked out this East DNA ligase 4 gene. So they knocked out this gene, so for which these nicks would not be translated, resulting in the DNA to remain broken, right? So but they observed that there was still, after deleting this gene, there was no lifespan reduction. So non-homologous enjoining was not a solid reason for lifespan reduction. This is just a evidence-based observation.
Okay. Fine, then it was okay. Another thing, do I have anything else to say? Okay, SIRTWIN2, sorry, not SIRTWIN, SIR2 knockout.
This is a bit confusing. Yes, sir. That since I was also initially confused that do SIRT genes code for SIRTWINs? No, it is not exactly like that. SIRT genes, they code for SIRTWIN-like proteins, but they function. Their function is exactly same as SIRTWINs in humans, but they are in fact they share some like
are they homologs by any chance?
Sort of, yes, they are homologs. But there are some differences in the amino acid composition as evolution has proceeded, but the basic function remains the same.
Did the authors check when they did SIR2 knockout, was there an increase or decrease in expression of SIR3 and 4?
No, sir, they didn't. No, sir, they didn't do anything like that. They never mentioned about it in the results section as well, or even in the discussion sections. But as far as I have understood the research paper, it is these SIR2, SIR3, and SIR4. These genes, they all have an effect on all of these 5 results, which I have actually tried to explain. But the thing is that the effect of one gene is dominant in one result as compared to the others. Like suppose, for example, if I consider the A and alpha mating type genes, this co-expression causes haploid sterility, causing reduced lifespan in the haploid cells. So in the case, it is the case of haploid yeast cells, right? So if we consider the case of haploid yeast cells, there the mating type co-expression causes reduction in lifespan. So there what happens, SIRT2 is also functioning. SIRT2 is the SIRT2 the absence of SIRT2 is also causing an important effect, but the absence of SIRT3 and SIRT4 is dominant over the effect of SIRT2. It is just that.
Okay. I think, Antik, I had only these questions. Thank you. Agradip, do you have anything to present?
I just wanted to show a paper that I presented. If you guys have like 10 minutes or so.
Yeah, sure. Please. Is it okay with everybody?
I'm turning off the presentation. Wait, sir. 1 minute. How to turn off? Okay. Yeah, this. Yeah.
Did this presentation get you into Heidelberg?
No.
Okay.
No.
I thought this is
This is
that one.
this is I don't know if you can see my screen or not.
Not yet, not yet. Now it's visible.
Yeah, so this was one of the papers that I presented here, I think in June or so. Don't ask me the details, but yeah. So this is what I meant when if you see here. So this is what I meant when I told about using like one or two figures per slide. So instead of like having everything together, you just separate each segment and you give a certain legend with it so that the audience is not overwhelmed. But you see the title is the same, so I am explaining the same thing, just the different figures with like each figure having its own inference. So yeah, this is how I separate the figures. That's what I told. I think Aditya and his another group member. I don't
Yes,
remember.
sir.
Shagnik.
Chagnik. So this is what I meant. So you, whenever you are transitioning into a next segment, just give a spacer slide. So even if this is like 25 slides long, all these are the parts of the same figure. So it will just take the same time as it would take to present everything in one particular slide. It just makes the thing like cleaner, and the audience will be more engaged, I think. And you can also use certain animations to highlight the certain areas that you want to uh, certain graphs. See if, for this case, I this is if these squares weren't there, this would have been particularly difficult to follow, but I use these green squares for these ones, the ones that highlight the green part, the SEABREVLPs, and the SMRNAs with the red one, so that people can easily follow this. So yeah, that's what I was telling about. And whenever there's a new technique, let's say the paper is explaining that, okay, we did this using this a new technique. So you can search Google or anywhere and you can get a quick summary, one figure which explains the technique so that the audience is not left in the blue that, oh, what this is. We don't know what this is. So yeah, so this was one of the new techniques that the paper had used, this ELISpot assay. And I will have to assume that nobody in my audience understands what an ELISpot assay is, even though there will be people who will know it, but there might be also people who don't know it. So I have to explain them a little bit about what an ELISpot assay is in one simple slide. And then I can show them the results of the ELISpot assay so that they can like relate, okay, this is what happens and this is the results. So, and this is the discussion. And I kind of use these pointers so that it's easier to follow which of the discussion is giving you a negative conclusion and which of them are the positive ones. So this is just like a quick view of my presentation. I hope you guys could follow. And if you have any questions, you can ask any of the panel members. We are available via LinkedIn, email. And yeah, it was nice having you guys present, and we presented. Hopefully, it was a good session. So yeah, from my end, it was wonderful. And yeah, all the best with your upcoming ventures, presentations, internships, whatever. Yeah, and that's all from my side. Thank you, guys.
Do you have anything to say? Anybody of you? Anybody from the student side? Something which you expected but didn't happen? Because we are at the end, finally. We just have one MCQ exam, of which half of the question is ready, half of which is remaining. I'll do it. We'll have it in the next week on Saturday. Anything do you have to ask to me or any of the panelists maybe? Nothing? Okay. I
No,
hope
sir.
we and people are not sleeping. Another thing, if you are giving just a lot of acknowledgements I have received, so in that, if you are giving acknowledgements in your presentations, make it, point wise. If you're acknowledging, like, let's say me in this case, then just keep my name and maybe the Department of Biotechnology and the HOD or something like that. You don't have to write statements, paragraphs in your presentation. It doesn't look good. While you write your thesis, the things are different. There you have to write the entire paragraph about acknowledgement. So that is one thing. It's okay, fine. I think we have all tabulated the results. It will be with you within a moment. We'll share it with you. And yeah, thank you very much. If it is fine with all of you, you can leave. And the panelists can stay actually.
Thank you,
Sir,
sir.
sir,
Thank you, sir.
Yes, yes.
In the next Saturday, when will be the exam time? At 3 p.m. or in morning?
11 a.m.
Okay, sir.
Okay, if that's all, you people can leave. We